Okay, good afternoon, everyone. I'm Roger Fielding, the Associate Center Director here at the HNRCA at Tufts, and I want to welcome you all to the kickoff of our spring HNRCA Monday seminar series. It's my pleasure to introduce today's speaker, Dr. Sarah Booth. Dr. Booth received her Ph.D. from McGill University in Montreal, Canada in 1992. And then joined the HNRCA. She has had several leadership roles at the HNRCA, both as Associate Center Director from 2019 to 2016. And since then, she has been our Center Director. And despite this large administrative and leadership burden, she has also maintained a very active research program in vitamin K and vitamin D nutrition. She is the lead of the vitamin K laboratory here at the HNRCA, which is the only laboratory in the US dedicated to the study of vitamin K nutrition, has developed the original assays for measuring plasma phyloquinone, which have become the standard for vitamin K assessment. I'm really excited to hear Sarah's seminar today on the role of vitamins D and K in the risk of Alzheimer's disease and dementia. Welcome, Dr. Booth.
Thank you, Roger. I'm just going to bring up my screen. Thank you very much, Roger, for the kind introduction. I hope everybody who is in Massachusetts were able to dig out safely and they have power. And I want to say a hearty Happy New Year to all of you who celebrate the Chinese New Year, because it's my understanding it is the New Year now in China with the time difference. So this is a really, really exciting talk for me to give today, because to me, this research is not about my work, but it is more about the team, the many, many people who contributed. And it also has made me really appreciate through this program the huge potential for collaboration within the center across Tufts, and certainly with collaborators such as my collaborators at Rush, the Rush Alzheimer's Disease Center and the Medical College. I wanted just to put everyone's name up because, as you can see, what I'm going to present today represents the contributions of many people, many trainees, many core unit managers, and many scientists. And also, it represents a collaboration with multiple talented people at Rush Medical Center. Finally, I just want to dedicate this talk to Martha Claire Morris, my collaborator, who unfortunately died during this, this project and was unable to see the fruition of all her efforts. I also really want to thank the funding agencies without whom this would not be possible. So let's talk about dementia. This is the latest forecasts of dementia were just released in the Lancet this month. And if you go to the 2019, the prevalence of dementia is about 50% for all women by the time that they have reached 95 years of age. And men, it is more in the vicinity of 40%. By 2050, we'll have the same prevalences of dementia among men and women. The difference is, as many of people in this audience realize, is that the demographic of older adults is accelerating exponentially globally. Therefore, dementia is going to be a monumental public health problem as we move forward as people age, because this is truly a disease that manifests itself primarily in ages 70 above. Now, one of the wonderful aspects of our collaboration with the Rush Alzheimer's Disease Center is they've really educated me and the rest of the team on what we are talking about when we're talking about dementias. And I think for many of us, particularly when we go from a nutrition perspective and we go into the field of dementia, we tend to think exclusively of Alzheimer's dementia. And as you see here depicted in the blue, Alzheimer's dementia pathology definitely increases with aging, but it's not the only pathology. And indeed, there's some other pathologies that are non-Alzheimer's disease, but the predominant number of pathologies as we age are mixed. They're a mixture of many, many types of dementia. And for those of us new to the field, it's a learning experience to appreciate that there are many, many reasons why we have cognitive decline as we age. So we decided to look at two nutrients for which there was biological plausibility in having a role in dementia and cognitive decline when present in low doses or low status in the body. One was vitamin D and one was vitamin K. We were very, very fortunate to get NIH funding to look at the two together. So for the purpose of my talk, we're going to separate the vitamin D and the vitamin K. So just to remind everybody about vitamin D metabolism, the active form of vitamin D is, of course, the hormone 1,25-dihydroxy-D3 or calcitriol. And the action of this hormone is to, when it reaches target tissues, is to affect a biological response that varies in accordance to the target tissue. The other point that I really want to emphasize here is, as you can see from this figure, vitamin D metabolism is highly regulated. There are many, many pathways for where excess or surplus can be oxidized and cleaved. It's a lot of feedback mechanisms. So this is a very tightly regulated pathway. For this reason, we do look primarily at 25-hydroxy-D3, which is the main circulating form that is converted by the 25-hydroxylase in liver from the parent form, the vitamin D3, which we can obtain from diet or we can obtain from solar exposure and production. The enzyme that is critical for the conversion of 25-hydroxy-D to 1,25-dihydroxy-D3 is this 1-alpha hydroxylase. And I'm pointing this out because it was really the discovery of that enzyme in many other tissues and organs that really was responsible for a lot of the explosion of vitamin D research. Until that point, the focus on vitamin D was for its still prime role, which is to facilitate calcium absorption. Very little calcium is absorbed in the absence of vitamin D, and calcium is so key to many of our biological functions. However, beyond the bone story, of which Bess Dawson-Hughes, who's part of this team, has clearly established the key roles of D and calcium, there was a lot of interest in what are other roles that vitamin D can play, including in the brain. So if you go into PubMed right now, there's about at least 100 papers a year that is looking at vitamin D status and cognitive function. It is known now that the 1,25-dihydroxylase, the 1-alpha hydroxylase is present in the brain. So the assumption is if that enzyme is there, the 25-hydroxy-D is being converted to the active hormone. It must be there because there's a role for the active hormone in the brain. Now, the earliest I can find any data that correlated serum 25-hydroxy-D with cognitive function came from 2007 in this small study by Neil Binkley and colleagues, in which he showed that increasing serum circulating levels of 25-hydroxy-D was associated with higher performance in the mini mental status examination. For those of you in the field of cognition, this is a very crude measure, so therefore this is a very surprising and encouraging result. There was some subsequent data that originated from Bess Dawson Hughes's lab in collaboration with one of her former graduate students, Jennifer Boel, that showed similar associations. So all very, very positive. However, I think we have to temper enthusiasm with a scan of all the data, and indeed it's very mixed. And the most recent data was from the VITAL study, in which adults were in a clinical trial. They were given vitamin D and or omega-3 fatty acids. Now, while not the primary outcome, cognitive change in cognition over a 2-year period of time, as measured by a battery of cognitive tests, they actually determined that there was no benefit of vitamin D on cognitive decline over 2 years. Now, it's beyond the scope of this seminar to debate the merits of measuring cognitive decline over 2 years using cognitive tests. But the point here that's very, very important is that there's a lot of mixed results in the literature. And it was in this context that we started our study. Now, one of the strengths of the center is that we are really good at analysis. And what we were really kind of dumbfounded was there were all these studies looking at serum vitamin D, which 25-hydroxy-D, and cognitive decline in humans, but nobody at the time had even ventured into looking at the brain. Was there even vitamin D in the brain? Was the 25-hydroxy-D simply a biomarker of something else? So Cheyenne Fu and our center, in collaboration with Greg Dolan-Lakowski, spent 5 years developing a very robust mass spec assay at the HNRCA to measure vitamin D in the metabolites. This is a busy slide, but I did it on purpose. I wanted to demonstrate that there was a lot of rigor in this assay, including validation, replication, and what we found when we were doing the assay is the predominant form that appeared in the brain was the 25-hydroxy-D. But in the method development, we were only using one human brain. So with this assay, we proposed to participate in the Rush Memory and Aging Project, the MAP. This is an amazing cohort, and I encourage anybody who is interested in the role of nutrients and aging of the brain to really look at these cohorts, because they're so exquisitely executed and they just have a wealth of potential to answer many questions. So this is a project where they recruit people. It started in 1997, my apologies. They recruit people primarily from residential centers in the Chicago area. They do baseline evaluations. People cannot demonstrate any cognitive impairment at the time of enrollment. They do a thorough clinical evaluation. They do many, many questionnaires. When those individuals die, they have signed a consent form that their brain and spinal fluid, cerebral spinal fluid, be donated for autopsy and pathology. These people have multiple follow-ups every 1 to 2 years until the time of death. So this is the group we worked with. By the time that we would receive brain samples following the autopsy, the average age of these individuals was 92, and it was a predominantly white population. There was very little representation of underrepresented minorities, and it was about 2/3 women, 1/3 men. So what we did is our original plan was to analyze 4 sections in brain samples from 500 people. We wanted to look at the anterior watershed, the cerebellum, the mid-frontal and mid-temporal cortex. I put this up here because I just want everyone to appreciate the work that Cheyenne and our students and Greg did. This represents 2,000 analyses on the mass spectrometer. And what we found in these analyses is that more than half of the people did not have any detectable 125-dihydroxy-D in any of the sections examined. And there was very little parent D, or the type that you would get from in the diet. So whereas everybody had some 25 hydroxy D in the brain samples, so for the purpose of these analysis, we're just going to focus on 25 hydroxy D. Now, in this analysis, actually, it was another scientist in our lab, Kyla Shea, proposed we just do some sensitivity analysis because we were analyzing brains that were collected as far back as 13, 14 years ago. And nobody had ever measured vitamin D in the brain. We were the first ones. And what we found was that when you collected samples as far back as 6 years from the time of death, the vitamin D concentrations actually dropped significantly. Now, one could argue, well, maybe it was related to the supplement use but all these people, when we control for supplements, it did not explain this. So we realized the vitamin D is not stable in brain. And as a result, we basically had to discard these data for the subsequent analyses, which, let me tell you, is very painful because it was half our sample. We focused our analysis on brains that were collected we analyzed up to 6 years from death. And I think this is an important lesson for all of us when we do freezer studies, is to conduct sensitivity analysis to verify that our nutrients are stable. So we're going to start with cognitive function. And these tables are a combination of the work that Ji Fan and Catherine did in the biostatistics core. And being the thorough individuals they are, they've given us some notes to orient us. So we have the global cognitive function and the slope of cognition. And what these are values in giving us an indication of the individual's cognition prior to death. And so the higher the cognitive function, the higher the value, the higher the cognitive function. So you would predict if high vitamin D confer protective effect against cognitive decline, there would be a positive association. And indeed, that's what we saw. We saw significant positive correlations between the 25 hydroxy D in the brain and these cognitive functions, regardless of where we measured the vitamin D. In other words, all 4 sections of the brain, we saw the same correlation. The flip is that when you do something called a cognitive diagnosis, the higher that measure is, the higher the measure, the more disease burden. So if vitamin D has a protective effect, you would predict an inverse relationship, and indeed we saw that across all sections of the brain. So this is cool, right? We're looking at cognitive function in these individuals. We've got half the samples we originally started with. We had about 280 people at this point after we corrected for the instability in the brain over time. And what we found was the more vitamin K in the brain, the higher the cognitive function prior to death. So this was really encouraging. Then we looked at the vitamin D plasma measures. Now, one thing that I didn't put up here is that but important to know is the 25 hydroxy D measures in the blood correlated with the brain. So you would expect to see exactly the same results, but in fact, we did not see it at all. And when you looked at the 25 hydroxy D, we really saw nothing when it came to cognition, which was a little bit surprising and a little bit disappointing, but very consistent, for example, with the VITAL study. Now, just one point for those of you not familiar with the vitamin D metabolism. Vitamin D primarily in circulation is bound. It's primarily bound to the vitamin D binding protein, a lesser degree bound to albumin. And so there's also a very, very small amount, and it's in the single percentages of the vitamin D in circulation is free. It's not bound to these proteins. And there's a theory that the 25 hydroxy D, because it's not bound, may actually be more bioavailable to the brain and be able to access past the blood-brain barrier. Now, what was really fantastic is during the time we did this study, the first commercial assay to directly measure free 25-hydroxy-D became available. And so we were able to obtain free 25-hydroxy-D measures. Now, it was a bit of a tease, I'm going to say, because we had this, when you looked at global function, there was a trend towards higher free 25-hydroxy-D, higher global function. We had in the right direction that the higher the free 25-hydroxy-D, the lower the disease burden. However, these don't reach significance, so we don't know if this is a spurious finding or if we need to increase the numbers. But this is very encouraging to us that any future studies we do will need to include the free 25-hydroxy-D. Now, one of the beauties and the wonders of the RushMap study is they have so much neuropathology. This is where the Rush Medical Center, the Rush Alzheimer's Disease Center really excels. And this is a really, really busy slide, but I wanted to put it up there to just give you a glimpse of the breadth of outcome measures available. Now, when we looked at the 25-hydroxy-D in the brain, and we looked at all the measures of pathology, be it global pathology, measures of Alzheimer's, plaques, tangles, etc., there was absolutely nothing with the 25-hydroxy-D in brain, which was very disappointing, particularly as we had seen the 25-hydroxy-D in brain associated with the cognitive. So then when we went over to the plasma markers, we had a similar picture. There just really wasn't very much with the vitamin D and the neuropathology, with one exception, and that is that the higher the 25-hydroxy-D, the lower the chronic microinfarcts, which is very, very interesting observation, given there are some data indicating vitamin D has a role in blood pressure. And again, this is one of many, many comparisons. So it could be completely spurious, or it could mean that there may be a role of vitamin D in more of the vascular pathologies, which was not the focus of this project, but we hope to be pursuing in the future. So in discussion with our collaborators at Rush Medical Center, the question was, how do we interpret these data? And this has been a real lesson to me as a scientist, because when we look at nutrients, we tend to look at these linear chains of causality. So let's take 25-hydroxy-D. You know, 25-hydroxy-D somehow, when it's low, can result in some disruption of amyloids or tangles, which leads to neurodegeneration, which leads to dementia, which explains these observations of 25-hydroxy-D in circulation being inversely correlated with cognitive decline. But maybe that's not what we're looking at. Maybe, for example, if we took the observation that the 25-hydroxy-D in the brain was linked to the chronic microinfarcts, maybe it was the 25-hydroxy-D influences vascular pathology, which affects dementia. Remember that first slide, it's not just Alzheimer's, which you would capture with the amyloids and tangles. It could be other pathologies that would also influence cognitive performance. It could be that there's some risk factor that has a mechanisms we don't know about, but it's more likely that it is modulating these relationships. It's very, very possible, and in the experience of our collaborators at the Rush Alzheimer's Disease Center, many, many nutrients fall into this category of risk factor 3. It doesn't have a direct line of causality. Instead, it modulates the other risk factors that result in dementia. And so right now, our thoughts is that this is where vitamin D may be influencing either the vascular pathology or the Alzheimer's dementia, which results in dementia. But clearly, we need to do more studies, and we need to increase our sample size, given our sample size was reduced by 50%. So in terms of the vitamin D, we concluded that there were higher postmortem brain levels of the 25-hydroxy-D were associated with better cognitive function that was measured prior to death, but they weren't associated with any neuropathology measured. The higher circulating 25-hydroxy-D levels before death were not associated with cognitive function, but were associated with lower microinfarcts. Indeed, the last slide, I made a little bit of mistake when I said the brain levels were associated with the infarcts. It was the circulating forms. So we believe we need to expand these studies. I would argue that we've got some great preliminary data, but we need to increase the sample size. We were originally powered for 500, but because of the instability of the vitamin D in brains, we reduced that to about 280. We also want to consider vascular pathologies, which were not addressed in this study. So now I'm going to shift gears to vitamin K metabolism. Sorry, I'm just going to take a... Okay. So just to remind everybody, vitamin K actually is a generic term, and it refers to 11 forms of vitamin K. And the vitamin K forms all share a naphthoquinone ring, and they all, but they vary in their side chain by length and degree of saturation. We focus in the diet on phyloquinone, which is very prevalent in green leafy vegetables and plant oils. We now know the menaquinones, which are also produced by gut bacteria, but are very prevalent in the food supply in the form of fermented foods and foods that such as cheese, and we have them see it in milk, and we also see it in a lot of cured meats. Menaquinone 4, I just want to emphasize, is an interesting form that we will be discussing later. But the important thing to know about menaquinone is it has exactly the same length of a side chain as phyloquinone, has the same naphthoquinone ring. The only difference are these 3 double bonds. So we have just determined, this was the work of one of our graduate students who's just graduated and gone on to do a prestigious postdoc at NIH, Jesse Ellis. What we have now discovered is that all forms of vitamin K, be they phyloquinone, menaquinone 7, 9, 10, they all metabolize to menaquinone 4. So we're still struggling with what this means, but what we now know is the naphthoquinone ring of phyloquinone, the side chain is cleaved off. We do not know how it's cleaved off. We do not know the enzyme, but the resultant form is menadione. We don't know where this happens. We don't know. Menadione is a highly reactive substance, so we don't know if this happens in the intestine, but the data are indicating now that that could be the predominant site. And then there's a side chain added, a prenylation, that is the critical enzyme for this is the ubiad D1, which is critical in the critical enzyme in the cholesterol synthesis pathway, and it's the side chain that forms menaquinone 4. We don't know why this reaction happens, what the difference is between phyloquinone, but this becomes very important in understanding the data we found for vitamin K. The one thing that is really important to note is it has been well documented using stable isotopes that in mammals, menaquinone 4 is the preferred form of vitamin K in the brain. So in these 2 studies done by Okano, what they did is they found that when you gave either phyloquinone or menadione in the brain, it was the menaquinone 4 that accumulated. So it's a critical metabolite. It's going to raise more questions at this point, I think, than I can provide answers today. So one of the issues that we are focused on in our lab with vitamin K is, do we eat enough vitamin K in the diet? In a recent analysis by our former students, Stephanie Harshman, using NHANES 2011-12 data, here this bar is the adequate intake, which is the median intake that's determined of vitamin K as determined in the NHANES 3 that took place in the early 1990s. You would expect, if there's been no change in the food intakes across the decades, that half of the columns would be red below and half would be yellow above, because this was determined as the median intake. Women's appear to be consuming slightly more vitamin K than they did in the 1990s, but older women, those over 70, definitely have consumed less vitamin K than they did prior. Compared to the younger age groups. So what you see is about 45% of all older women do not meet the adequate intake. In contrast, older men are consuming less vitamin K as recorded in previous decades, and about almost 70% of older men do not meet the adequate intake. So it's definitely a nutrient that is not consumed and as much as we would have as recommended. And it would suggest that a lot of people are not meeting the dietary guidelines for Americans, as phyloquinone is easily obtainable in healthy foods such as plants and plant oils. So the purpose of the vitamin K is it's an enzyme. Whoops, sorry, this is timed. Is an enzyme or helper cofactor. The vitamin K enters into a cycle that is reduced. The hydronaphthoquinone form is the active form, and this drives this reaction for the carboxylase. So this vitamin K dependent glutamate carboxylase enzyme is responsible for carboxylating glutamic acid residues in certain vitamin K dependent proteins. So in essence, what it does is the specific glutamic acid residues in certain proteins, the glutamic acid has only one carboxyl group. In the presence of the active form of vitamin K in this enzyme, all it does is adds another carboxyl group. And that creates a conformational change to the protein to allow them to bind to calcium or what other compound that the individual proteins are binding to. The active form is recycled and then further reduced back to the same form you saw in the diet, and this is continually recycled. So the only known role right now is as an enzyme cofactor, and the key beneficiaries are the vitamin K dependent proteins. Now there's about 17 vitamin K dependent proteins, but relevant to our discussion today, GAS6, one of them, has been measured in the brain, as has protein S. GLA-rich protein and matrix GLA protein, which are found in circulation in the vascular smooth muscle cells, appear to be regulating calcification. The protein S is a critical protein in the coagulation system. GAS6, the role of this is still under investigation. But I told you before that phyloquinone is converted to menaquinone 4, and we can't figure out why. Because menaquinone 4 is perfectly capable of carboxylating these proteins, and its ability to carboxylate these proteins is the same as phyloquinone. So the question is, why are we undergoing this expensive biochemical transformation to form menaquinone 4? And the short answer is we don't know. One theory, though, is that it's a ligand for SXR, which would be PXR in mice, and that this ligand forms this steroid and xenobiotic receptor is required for gene expression of proteins involved in bone, and other different functions. However, this is still very, very speculative. And the bottom line is we just simply do not know what roles menaquinone 4 has that cannot be achieved by phylloquinone or the other menaquinones. Now we know all menaquinones form it. So let's shift gears and go back to the MAP study. So when we analyze these 4 brain samples, again, to reorient you, the anterior watershed, the cerebellum, the mid-frontal and mid-temporal cortex for vitamin K, what we found was that the predominant form was menaquinone 4, which was consistent with the animal data. And indeed, only 3% of all the 500 brains we looked at did not have any menaquinone 4. In contrast, there was almost no phylloquinone in brain, which we were very surprised. About 83% had no detectable phylloquinone. So for the purposes of these analyses, we just focused on menaquinone 4. Now, Kyla once again suggested we did sensitivity analysis, and indeed we found that vitamin K is not stable indefinitely in the brain. Now, in contrast to the vitamin D, we found that vitamin K is fairly stable until maybe 8 to 9 years. It's statistically lower by 9 years. So we made the decision to only include brains from 8 years from the time of collection to analysis forward. And this way we would minimize any confounding by instability and hence low vitamin K that was related to storage and not to actual status. So with that, we had slightly more brain samples to analyze than we did with the vitamin D. We had around 320. Again, like the vitamin D data, we looked at the 4 sections and we looked at cognitive function and summary. And very similar, in fact, even more so than the vitamin D, we found that the higher the menaquinone 4 in the brain, the higher the cognitive function. And it was all statistically significant. We found that the higher the MK4, the lower the cognitive summary, hence less disease burden. So this was really exciting for us. Then we went to the plasma markers and we saw exactly the same thing. Now there's a couple of points that I want to say here. The phylloquinone in the blood was not correlated in any way, shape, or form with the MK4 concentrations in the brain, which was a bit of a buzzkill for us because that means that what we measure in the blood would not be able to be used as a biomarker for the brain concentrations. However, we also are very sensitive to the fact that vitamin K in the blood does change in accordance to a dietary shift fairly quickly. So we did both the second visit ever of the participants in the MAP study and the last blood draw. And we looked at them separately and then we combined them because we wanted to see if there was any temporal effect, but actually the data were the same and the bloods collected in the second visit and the last were statistically the same, which gave us a sense that it was more robust. Now, it's not reflecting what's in the brain, so our interpretation is that it's probably reflecting a healthy lifestyle, which even though we adjust for the DASH score, we may not be able to differentiate healthy lifestyle from the vitamin K per se. Again, all speculation. Now, when we looked at the vitamin K plasma markers, we saw the cognitive work, and like the cognitive associations, like the vitamin D, we saw nothing. Here we also threw in one of the vitamin K dependent proteins, matrix GLA protein, and there was nothing. We just did not see an association between the vitamin K plasma markers and neuropathology. But we then went into the neuropathology and the MK4 brain, and this is where we got really excited. So in terms of the global pathology, the higher the MK4 in brain, the lower the overall pathology in these brains. It's significant in the anterior watershed. It was a trend in the other two sections. The Brack stage is an indicator of Alzheimer's disease. And we found the higher the MK4 in the brain, the lower the severity of Alzheimer's disease. But we didn't see anything with the plaques, which is one of the hallmarks of Alzheimer's disease, but we did see a very consistent pattern with tangles and, interestingly, Lewy bodies, and nothing with the infarcts. But this is exciting to us because we have some very consistent findings here. And so when we're looking at this, when we look at the MK4, we definitely are not seeing anything related to amyloids, but we do appear to be seeing something with tangles. And we don't know if this is resulting in the neurodegeneration and the dementia, or is it another factor, or is the MK4 here modulating? But given the neuropathology in the brain levels, we believe that it's going for tangles and possibly also the neurodegeneration. Again, this is a correlative study. And because we are at the HNRCA, we had these really exciting findings, and I was jumping up and down because we didn't expect to see anything with the vitamin K. So I went to our colleagues in the neuroscience lab and I said, Tong, we find this cool stuff, but I don't know what it means. And it's also a correlative study. So Tong and Cheyenne said, well, let's just start with a basic mouse study. So they took some wild-type mice, black 6, no genetic manipulation. It's a very simple study. And they aged them. And half the group was low vitamin K, and half the group was a controlled diet. And then we had females and males. They did behavioral tests over at the Tufts Medical School, where they have a fantastic behavioral core. And we are going to, Tong did the Birdue injections so we can look at neurogenesis. Now, one of the big problems we had was the males in the low vitamin K group, half of them died. And with collaboration with Sheldon and Alan, we also did discover we have some visual problems in these animals. We just don't know what they are yet, because these are pretty well hot off the press. Now, what was really exciting was we have the behavioral data, and what we're seeing when you look at the novel object test, which is a recognition memory test, what we're finding, I just want to draw your attention to the fact that the males is where we're seeing all the action, and the those who get the regular vitamin K, their recognition memory is fine. There's impaired recognition memory in the males that are being fed low K. In the Morris Water Maze, which is a spatial learning and memory test, again, drawing your attention to the low vitamin K males, it takes them longer to figure out where the platform is. These are really preliminary data, but it shows the strength of translational science that we can do here at the HNRCA. We get a correlative finding, we can then go and do animal studies based on then the analysis of neurogenesis. My collaborator in Texas is going to be looking at GAS6 expression in the brains. We're going to look at vitamin K. Based on that, that will give us clues as to what then to look next in the future studies with the RASH map study. So in conclusion, what we found with respect to vitamin K is we had higher postmortem brain levels, metaquinone 4, and those were we found those were associated with better cognitive function. We also found that the postmortem brain levels were associated with lower global AD pathology, specifically fewer neurofibrillary tangles, and they were also related to fewer Lewy bodies. The higher circulating phyloquinone levels before death were associated with better cognitive function, similar to that which we saw in vitamin D. but they're not associated with the neuropathology study to date. And we believe that the phyloquinone levels are probably more of a marker of a healthy diet that's creating some kind of resilience in the process to reduce the overall cognitive decline. These are all correlative studies. We need to do mechanistic studies to understand the underlying mechanisms and evaluate causation. And I just feel we're just so fortunate that we were able to, within our own center, initiate the first studies of this kind to really see if there's something there and start understanding the hows and the whys. So again, I promised that I would do it in 45 minutes, and I want to thank you again for listening to me. And I just want to thank all our collaborators because this has been a journey with many people. So thank you.
Thank you very much, Sarah, for a really stimulating and really interesting talk. And there's a lot of great work here that's advancing this field of nutrition and neurocognition. And I think it's really work. Number of questions already coming over. Let me start with one from Ryan Snodgrass. With regard to vitamin D3 metabolism and activity in brain, he's asking which cells express CYP27B1 and which are targets of calcitriol. In other words, the cells that express vitamin D, VDR, vitamin D receptor.
Oh dear, I think I need to defer to Bess Dawson Hughes for the answer for that. I apologize. I don't know which cells in brain. I'm not current with that that literature. I don't know if Bess is on the call, if she would be able to answer.
Oh, she can. Well, we'll come back to that
My
one.
apologies, Ryan.
Here, and then Barbara Shookett-Hale asked for the vitamin, and this was something I was wondering about as well. For the vitamin D study, have you thought about breaking the overall cognitive score into cognitive domains? For example, cognitive measures of executive function have been found to be dependent on vascular function, while measures of memory function are not related to vascular function. Any thoughts about domains of cognition, Sarah?
Yeah, so Barbara, this is a really good question. I defer to my collaborators at Rush Medical Center who have extensive experience in this, and they have developed more, they prefer the scores as opposed to the individual cognitive tests. I think their main concern, and I know that Catherine definitely embraces this, is multiple testing. I mean, you're just looking at so many different things. So my understanding is that we're really dealing with the scores, and I defer to the experts on that they feel that's a more robust way to evaluate.
Yeah, good point. Alan Taylor asks, if I heard your description correctly, you indicated that in the brain there is hydroxy vitamin D. I think he means the 25 hydroxy D, but no parent compound nor dihydroxy vitamin D. And how do we understand that, or why do you think that is, I guess?
Oh, absolutely. So Alan, actually, we did measure in some people, there was parent vitamin D. We even measured the vitamin D2 form. We measured the D3. We also did measure the 125 dihydroxy D, the calcitriol. It's just not everybody had all those forms. And one of the challenges we have, which I'm sure you all relate to, those of you who work with measuring tissue levels, is at the time, the lower limit of detection of the Q-trap didn't allow us the confidence to really include those lower numbers in the analysis. We were a little worried that they were spurious events. What we are hoping, we're close to getting this grant, another grant, the grant renewed. We have a brand new Q trap at the HNRCA, which has magnitudes, high levels, more sensitivity. And we believe when we use the new Q trap, we will be able to dive deep into the 125 dihydroxy D levels, which I agree are probably the more interesting form. It's just we didn't have the sensitivity of the assay. We're continually pushing the limits of technology. I do believe Bess is joining us to give an answer. Yep, Bess, would you like to answer the question on the SIPs?
Yes, you can unmute yourself since you've been made a panelist now.
Oh, okay. Can you hear me now?
Please.
Oh, sorry. You'd have to repeat that question. I was busy writing a new question for Sarah, which
Okay. Trying to go up to the, here we go. Hold on. Just having a little.
Bess, with regards to vitamin D.
Here we go.
Oh, go ahead, Roger.
Yeah, with regard to VD3 metabolism and activity in the brain, which cells express CYP27B1 and which are targets of calcitriol and cells that express the vitamin D receptor, the VDR?
I don't know the answer to that for brain tissue, and I'm not sure that it is known, actually.
Okay, great. Thank you, Bess. And Alan Taylor, I'll go to a new question. Alan has another question about supplements, but Chris Wiley asks, I understand that this is a bit early, but could the difference between human blood and brain vitamin K levels suggest that consumption of vitamin K may not be as promising as an intervention as finding a way to get the K into the brain?
Chris, I'm not sure that's true. It's a good question, but I don't think that may be true. I think that the blood levels reflect very much what we have just consumed in the diet. But as I hope to convey to you, that vitamin K form that we obtain from the diet is then metabolized to form vitamin MK4, which goes in the brain. We know so little about how it gets metabolized and transported that we don't really have a good sense of the different steps. What I think more of, instead of saying that the consumption of vitamin K may not be promising, because it is, because in our animal studies using stable isotopes, if you decrease the vitamin K intake in the diet, the menaquinone 4 levels eventually go down. I just think it's more highly regulated than we appreciated before. And I think it's not a one-to-one linear relationship, but certainly the animal data indicate that you need to eat it in the diet to get it in the brain.
And there are some other questions coming in over the chat, sort of like about, would vitamin K supplements benefit someone over 60 and what would be the optimal form? Is there anything you can say about that at this point, Sarah?
Sure. I am so not in favor of advocating supplements based on an observational study. These are just really exciting, but we don't know how much one needs to eat to get the optimal amount of vitamin K in the brain. And quite frankly, looking at the data very crudely, I would argue that we're seeing the people with the pathology at the very low intakes of vitamin K, the very low blood level, brain levels of vitamin K. And I would argue if everyone strive for a healthy diet as advocated, for example, the dietary guidelines for Americans, they would do just fine. It's just most people do not like to eat green leafy vegetables and healthy foods such as that. So yeah.
Great. And Jerry Combs is asking that he's guessing your brain samples were stored without antioxidants. If so, the losses of 25 HCC and K that you saw were likely due to oxidative degradation. Which products should be present? If you can find them, you will be able to reconstitute the original levels, which would enable you to use more of your specimens. I guess it's maybe using the degraded process products as measures.
Yeah, that's a really good question, Jerry. And I suspect that could be the cause of the degradation. Unfortunately, the MAP study was not designed exclusively to measure nutrients in the brain. And so the protocols for the pathology and brain preservation are really being optimized for many other questions and primarily related to the pathology as opposed to the nutrient levels. So it's one of the wonders of collaboration. You get access to wonderful samples. The downside is you don't get much of a say in the protocol.
We have another question, Sarah, from Joel Mason. Congratulations on these very nice studies in the MAP cohort. If there is one hydroxylase in the brain, why is there no 125 dihydroxy-D in the brain? And then related to that, what is the functional meaning of the 25D in the brain, since it is perceived to either be non or minimally biologically active? Are you thinking that there may be a novel biological action of 25D? So lots of questions in there, but.
Thank you, Joel. You like to challenge me. It's an interesting way of thinking of it. My gut instinct right now is it's more of a numbers game. I think, as I mentioned earlier, I think the concentrations of the 25 hydroxy-D are higher but the lower limit of detection for the 125 dihydroxy-D are unfortunately too low for us to detect. I think that I would rather be redoing this with the new Q-trap before I could answer that question, actually.
Great. I'm getting questions from the Q&A and the chat, so I'm trying to go back and forth. If anybody has new questions, try to use the Q&A because I'm not that talented to jump back and forth here. But anyway, Emily Price asked, wonderful presentation. I was curious, in the MAP study, did any of the participants supplement with vitamin D or K? Was that measured or controlled for?
Very good question, Emily. Yes, under the direction of Martha Claire Morris, they do complete food frequency questionnaires in this cohort. We did control for the vitamin D supplement use. Vitamin K is it's very unusual for people to be consuming vitamin K in a supplement form. If it was in a multivitamin, they're usually between 10 and 25 micrograms per tablet, and it really wouldn't contribute. So I do not believe we control for vitamin K simply because people don't take vitamin K supplements, but we definitely did for vitamin D, and we also controlled for season.
And then any insights on why so many of the male mice died that were on the low vitamin K diet? Was it related to low K or some other cause?
Yes. So there's this really interesting phenomena that we can't explain yet, and I'm hoping there's students in the audience who would be inspired to study it. We find that there is a very, very profound sex-specific effect of vitamin K deficiency in the mice. I don't think we push the dietary extremes in humans to see it in humans, but males, it's well documented. If you remove much of the vitamin K in the diet, the males will become deficient within 2 weeks, whereas females can go on for months. So I think in the study with that Tong and Cheyenne did, it was an older group, and they were very deficient and they died, but it wasn't one reason they died. So that's they just are not very healthy when they're on low vitamin K diet. So that's why it's a sex-specific observation.
Great. And Danielle Calhoun asks, again, wonderful talk, Sarah, on some very niche and important research. Given the role of vitamin D in regulating immune response, Have you looked into or plan on looking at associations of brain or plasma 25-hydroxy-D with neuroinflammatory markers associated with neurodegenerative disease pathology?
Great question, Danielle, who just sent me a manuscript to review on neuroinflammatory marker. It's a great question. And indeed, Danielle, we're hoping to renew our current grant to expand into vascular pathology, because I see somebody else ask about the vascular pathology, and we would like to be including neuroinflammatory markers. All of these are available in the RUSH study. I should also mention that one observation I see Bess asked, the VITAL study, yes, Bess, you are correct that the average vitamin D in circulation for VITAL was 30 nanograms per ml, so they were replete. This is not the case with the MAP study. I think it was in the low 20s. So it's a group that has lower vitamin D status. But in the VITAL study, what's interesting is when they did a subgroup analysis, they found that Blacks were more likely to have more cognitive decline if they had a low vitamin D. So what we are proposing in future studies is to include a second cohort from the Rush Medical Center, which is the minority aging research study, which would include a more representative population. So lots of exciting things happening in the future.
Well, maybe if we have time for a couple of more questions, Sarah, is that okay?
Sure,
Okay.
sure.
Great. So Jean-Dong Wang asks, in your study, the higher brain MK4 levels were associated with lower global AD pathological lesions. Have you taken a look at other nutrients such as carotenoids, vitamin E and D in the same study?
Good question, Jean-Dong. We only looked at vitamin D and vitamin K, but Martha Claire Morris actually did look at carotenoids, and she also looked at vitamin E. So there are multiple studies from that group looking at various nutrients in the brain.
Okay, Thomas Beater asks and says, exciting work, Sarah, regarding the dissociation of brain and plasma levels of 25-hydroxy-D. Could this be due to differences in the expression of 25-hydroxylase or other processing enzymes across the brains of individuals?
I really can't. That's an interesting question. I really don't know, because I don't think it would be, because I think that what we're measuring in the brain is probably coming in as 25-hydroxy-D via circulation. I mean, that would imply that the parent D was getting converted in the brain. And I think it's more the 25-hydroxy-D to 1,25-dihydroxy-D. It's an intriguing question. I really don't have an answer, Tomas, sorry.
And as a follow-up to that from Tomas, in relationship to the vitamin K data on Lewy body abundance, does the RUSH cohort include patients with Parkinson's or Lewy body dementia? These could be interesting associations.
Yeah, thank you for that question, Tomas. So if you recall, right at the beginning, there was that beautiful, elegant prevalence of the different pathologies. And indeed, the RUSH Alzheimer's Disease Center has really focused on multiple pathologies, including Parkinson's or Lewy body dementia. Certainly the Lewy body association got a lot of excitement among the group, but I have been cautioned to be very careful about assuming Lewy body relates to Parkinson's, given that there's now an Alzheimer's, Parkinson's, Lewy bodies type dementia. But it is very interesting, and I agree with you, it's something that we really need to pursue.
Okay, and one final question from Alice Lichtenstein. Do you think the 25-hydroxy-D concentration association with cognitive function in the brain is a marker, could be a marker for a different putative factor?
I do, Alice. I think that the 25-hydroxy-D it's probably conferring some form of resilience. I think that would be the most plausible hypothesis, but I think we need to explore a lot more. These are unpublished data, and so far we haven't seen any other data yet in the brain with vitamin D. So I think there's a lot to be done, but I sort of think that it's probably related more to resilience than causality.
Okay, I think that covers all of the questions. Again, thank you for a very interactive question and answer period, Sarah, and thank you all who attended today's seminar. We look forward to having you join us for future seminars going forward. Thank you very much.
Thank you, everybody.